So as many of you already know, the heart disease is #1 cause of death in United States because the UAB is very well known in studying cardiovascular studies. Over 600,000 people are dead for the heart disease every year. Most studies in cardiovascular studies are for adult heart disease, such as atherosclerosis or valve calcification. But although the 1% of newborn babies are suffering from various types of congenital heart disease, congenital heart disease is less studied compared to adult heart disease. So our group is using a zebrafish as an animal model to study heart disease. I guess some people probably know how zebrafish looks like, but somebody probably not, right? So this is a zebrafish. So that's why people call the fish as a zebrafish. But yeah, but just kidding. This is actually the zebrafish. So you can find these zebrafish easily find the local pet store, such as PetSmart or Petco. So can you distinguish which one is a male and which one is female? Okay, for human, so usually woman has a lighter and brighter skin compared to the man, right? So it's the same as zebrafish. So the zebrafish, this one is a female. So female zebrafish has a shiny skin and brighter skin, shiny and brighter skin compared to the male. But zebrafish female has a lot of eggs on their belly. So it has more like curvy shape of the body compared to streamlined shape of the male zebrafish. So zebrafish is genetically tractable model to study congenital heart disease because a large number of cardiac genes are conserved between humans and zebrafish. When these genes are mutated, it associates with a phenotype that is related to the human heart disease. For example, TNNT2 gene is mutated in zebrafish, has a poor cardiac contractility. And this zebrafish is studied for human heart failure, such as dilated cardiomyopathy or hypertrophic cardiomyopathy. Thus, zebrafish is powerful animal model in translational medicine. Also, there are many other advantages. They are easy to culture. They're very easy to culture. So we can simply put one male and one female in the breeding tank overnight. And next day, they produce hundreds of embryos. And the organ system is developing very fast. So for heart, it takes only about 5 days to develop to have atrium and ventricle with a valve system. Furthermore, zebrafish is optically transparent, so it allows us for in vivo optical imaging and visualization of the cardiac structure or the brain structure or the vascular structure. So, and then it is genetically tractable model as mentioned earlier. So there's a lot of transgenic lines or mutant line already in the market, so we can access them easily. And the zebrafish society people are try to be, get together very nicely. So whenever we ask them to share their transgenic line or mutant line, they're willing to share each other's. So it's nice to have a zebrafish as an animal model to study cardiac development or heart degeneration, whatever cardiovascular disease. While the human heart has 4 chambers, 2 ventricles and 2 atria, zebrafish heart only has 1 atrium and 1 ventricle. However, their ECG is similar to the human, and so we can see the P wave and QRS complex and T wave. And also, we can observe the mechanical E wave and A wave during ventricular filling from Doppler ultrasound. Therefore, zebrafish cardiac physiology is similar to human physiology, although their anatomy is quite different. So we use zebrafish as an animal model to study cardiac trabeculation in the aspect of the developmental biomechanics. Okay, let me introduce what trabeculae is. So somebody probably doesn't know what cardiac trabeculae is. So cardiac trabeculae is irregular bundles of myocardium that project into the inner ventricular wall into the cavity. It is clinically important if trabeculae are too much or too less. It causes impaired diastolic function or systolic function. Hypotrabeculated ventricle induce hypoplastic heart syndrome, which is undeveloped heart. So this figure shows the hypoplastic undeveloped heart. So their chamber is relatively smaller than, basically much smaller than healthy ventricle. And hypertrabeculated ventricle induce non-compaction cardiomyopathy. This is a hypertrabeculated ventricle. So since extratrabeculae invaginate into the ventricular cavity, it causes non-compaction cardiomyopathy, which is third most popular congenital heart disease in a pediatric patient. And we have previously revealed that trabeculation is closely related in Notch signaling. Notch signaling is important biological cell signaling, which involved in many developmental processes. So Notch signaling is also affects both myocardial differentiation and proliferation. However, it is not clearly known what biomechanical quantities affects the morphology of trabeculation. We hypothesize that hemodynamic shear stress induced by blood flow initiate the trabeculation with endocardial Notch signaling, and later cardiac contraction affects the myocardial notch to terminate the trabeculation to design the wave-like pattern of inner ventricle. So basically our main hypothesis of this project is that the combination of endocardial and myocardial notch signaling are required for proper cardiac trabeculation and cardiac function, ventricular function. So do you have any questions for my introduction part before moving into the technical part of my project? I just want to stop here first and then ask you guys if you have any question, feel free to ask me. Okay, good. So if you don't have any question, and that's good sign for me. So all of you guys understand what my research is, right? Okay. So let me introduce my imaging system, which is a light sheet microscopic system. So unlike other microscopes, the light laser source is transformed to the sheet-like structure and illuminates from the sides. So this is the top view during imaging. So only the 2D section, only thin 2D section is illuminated by laser from illumination objective lens. So the background from this side and this side can be significantly reduced because this side and this side are not illuminated at all. And then we can detect, we can image with the detection objective lens, which is positioned orthogonally here. So this is actual zebrafish mounted in light sheet microscopic system in our lab. So this animation shows how we image the zebrafish embryo in 3D and reconstruct it. So this 3D reconstruction seems very similar to the confocal microscope, right? However, compared to the confocal microscope, light sheet microscope has many advantages. The confocal microscope and light sheet microscope both can do the image sectioning, but the light sheet microscope is much faster than confocal. Confocal is point scanning based with a pinhole which blocks unnecessary light. So the original fluorescent signal has to be strong to be detected. And it is not suitable for imaging dynamic samples such as heart because it's a pixel, so pixel by pixel imaging. So when we image the dynamic sample such as on heart, so when we take an image at this morphology, it already deformed, right? So the confocal microscope is not suitable imaging modality to take a dynamic sample such as on heart. However, light sheet microscope is suitable for imaging dynamic sample with a fast scanning since it illuminates entire 2D plane at a time. So it doesn't, and then it doesn't block any light. It doesn't have any pinhole or something. So we can image with weak fluorescent signals. So that's the advantage of the light sheet microscope compared to the confocal. Although the light sheet microscope itself provides a superb the axial resolution, we further developed the super resolution techniques to improve the 3D voxel resolution. So the whole idea is to scan the sample with a little bit of angle, basically like 6 to 8 degrees of angle orthogonally. Then we reconstruct the 3D image with every 5 or 6 or 10 slide. So for here, so the schematic shows a 3D image of the red slide. So this is the whole stack. And we are taking only red slides. So do you see the red slides here? Here, 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 here. So we have a 3D reconstructed image of the red slide. And we have yellow slides. We take yellow slides and reconstruct the 3D. And then we take the green slides to reconstruct the 3D. So because of the orthogonal scanning and different imaging depths, right? So because of they contain different imaging depth information, and then a little bit of shifted spatial information. Every 3D reconstructed image. So this one, looks slightly different, but very similar, but slightly different. So we are using the iterative maximum likelihood estimation to resolve the resolution, interpolate the image among each 3D images. Then the voxel resolution our voxel resolution can be improved up to 5 times. So this DEF shows the point spread function, and each full width half max to show the resolution improvement of our super resolution technique. So this technique has been highlighted in Nature Photonics in 2019. So furthermore, we have integrated this technique with a multi-view fusion technique to bypass the tissue scattering issue. When we are imaging the large sample, so usually, if we are scanning with a laser, we can have a very clear view on this side, on this top view. But when we scan deeper, we are going to have a blurred image because of tissue scattering, right? So we have a good image of this, and then we rotate the sample 90 degrees, and then take the image of here. So we have a good image of this side, and then 90 degree rotation, and take the high resolution and clear image of this side, and rotate 90 degrees to have a clear image of this side. And then we are fuse them to have a good resolution, good quality of images. So this image shows the clear structure of the Hubeck cell and human thermal fibrosis 3D sprouting network with our multi-view super resolution, multi-view fusion and super resolution technique. Okay, so now we apply our technique into the zebrafish. Compared to the confocal microscope, this is confocal microscope image. Our multi-view super resolution technique provides clear structure of the zebrafish head. Compared to here, it's the same area, but we have a much better resolution, So in the meantime, we can image entire zebrafish body, and then after we zoom in, we can have a clear structure. We can have a clear fine structure of the eyes and vascular structures. So when we use the low magnification objective lens to see the whole body structure, then we cannot see the fine structure after we zooming in, right? But so that's why that's why we are using high magnification objective lens to see the fine structure. Then we cannot see the whole body. So the main advantage of this work is to circumvent this trade-off and catch both goals. Okay, let's circle back to cardiac image. So traditional microscope is limited to capture the dynamic motion of 3D structure like a heart. So we have developed a computational algorithm to reconstruct the 4D beating zebrafish heart, assuming this is a zebrafish heart. At first, we take several hundred of motion of the zebrafish heart at this focused layer. Then we also take another several hundred of images from 1 micron bottom. Okay. Then in vivo heart motion image will be like this, right? So heart is beating, and when we take the image of this Z2 layer, it also beats, right? Then we compare the images. We compare the image intensity between Z1 layer and Z2 layer to find the minimum pixel intensity difference and shift image to match the same cardiac contractions. Since the distance between Z1 layer and Z2 layer were set to 1 micron, we assume that the image intensity at the same cardiac contraction is almost same. So this is the result. Raw data shows on top before processing. So contraction periods at each Z layers are not consistent, right? So they're all different cardiac contractions from Z1 layer, Z2 layer, Z3, Z4, right? But after processing, after processing with the computation algorithm, the contraction period of each Z layer become consistent. So they are all in same cardiac contraction. This stack of sliced generate 3D image of each contraction time period, right? So this shows us 6 different consecutive time points in this slide. So finally, we have this 4D beating zebrafish heart image. So this 4D beating zebrafish heart image provide the boundaries to quantify the biomechanics. So, so this is, so far I introduced my imaging side. So do you have any questions for the imaging side before I'm moving into the mechanical analysis? Yeah, you can feel free to stop me anytime, it's fine. Okay, thank you. So now I want to introduce the hemodynamic analysis that we have used the 4D beating zebrafish heart. So previously, we segmented ventricular fluid domain from 4D beating zebrafish heart manually by hand. So these images are from wild type zebrafish heart in different views. So this is left, right, this is front and the back. So however, this manual segmentation takes a lot of time and we have to spend a lot of effort on it. So therefore, we have recently developed a deep learning technique to segment zebrafish heart automatically. So A and B shows the volumetric geometry of the diastolic and systolic stage of the heart cycle. Compared to the auto segmentation and ground truth image shows there is just a small segmentation discrepancy. So if you look at this, this yellow color is overlapping area, and you probably see a little bit of green side over here. So this green side, the green is our ground truth, basically the hand segmentation. So green is a hand segmented image, and the red one is auto segmented image. So although there's a little bit of segmentation discrepancy, however, our dice coefficient score is 0.95 with the standard deviation 0.02, which often indicates a good segmentation efficiency. So this video shows the auto segmented so this video So this video shows auto-segmented zebrafish heart with the previous our 4D beating zebrafish heart image. So furthermore, we have done little bit more processing to separate the atrium and ventricle. So this method relies on iterative process which use the erosion operator on 3D structure. So we have erode the structure to erode operator on the 3D structure of the inner volume until fractioning occurs. So now we have a fractioning occurs, right? So following this, dilation operation was applied to individual separated each cardiac chamber to recover the lost space. For that, we can do many cardiac mechanical analysis, like cardiac mechanical analysis, such as the volume change, and then we can quantify the ejection fractions, and then like end-diastolic volume and systolic volume. So this example shows the real-time volume change of the entire heart volume and the atrial volume change and the ventricular volume change in real time with auto-segmentation. Before we simulate the blood flow to quantify the shear stress, we have done genetic manipulation to lower wall shear stress, the hemodynamic shear stress. We inject the GATA1 morpholino inhibit the hematopoiesis. Therefore, so the red blood cell, no red blood cell is circulating in blood, which lowered both blood viscosity and wall shear stress, because the shear stress is a function of viscosity. And also we use the WEA mutant to inhibit atrial contraction. So the wall shear stress on ventricle was also significantly reduced because the velocity from atrium to ventricle is inhibited. So this image shows the entire ventricular view with the light sheet microscope. So blood flow is coming from atrium here and going in and out to the outflow tract. Okay, let's cut and open to see the heart inside. Here is the image. After we inject the GATA1 morpholino and use of the WEA mutant, trabeculation is significantly attenuated compared to control group. So control group has a nice looking trabecular network. It's visualized. And then our 4D fluid domain segmentation provides a cavity volume change in time. From this data, we quantify the volume change in one cardiac cycle in response to control and GATA1 morpholino and then WEA mutant. We observe that the ventricle with attenuated trabeculation has less contractility, right? So it has a less contractility compared to the control group. So control group has higher contractility compared to the GATA1 morpholino injected group and WEA mutant. From the simulation, in collaboration with Dr. Vijay Vedula at Columbia University, we can see the streamline of the blood and spatial temporal variation of the wall shear stress when heart is beating. So we see the shear stress of control is bigger than other two groups. So these are the quantified wall shear stress results. So area averaged wall shear stress is averaged entire ventricular wall shear stress change in one cardiac cycle, and time averaged wall shear stress is the average of one cardiac cycle of area averaged wall shear stress. And wall shear stress from control group has significantly higher than other two groups, which has a less trabeculate, right? It means the trabeculation may be related in magnitude of the wall shear stress. So this is the confocal microscope image, the 4-day post-fertilization zebrafish, basically 4 days old zebrafish. So which means the trabeculae is very active to form at this 4 days post-fertilization stage. So wild type with a high hemodynamic shear stress wild type with a high hemodynamic shear stress initiate the trabeculation, right? While Garawan morpholino injected zebrafish, which has a significantly lower shear stress, didn't initiate the trabeculation. Also, with using special transgenic zebrafish to visualize the cardiomyocyte proliferation, so called the FUCI system. So newly proliferated cardiomyocytes were presented at trabecular ridges. So newly proliferated cardiomyocytes are actually located in the majority of them are located in trabecular ridges. From this study, we revealed the hemodynamic shear stress initiate the trabeculation via endocardial notch signaling until 3 days post-fertilization. However, after initiation, notch signaling transit to the myocardial area to inhibit trabeculation. So next study, we will try to understand the mechanism of cardiac contraction, how the contraction related in myocardial notch signaling to inhibit trabeculation to create these trabecular ridges. So now we want to understand how myocardial deformation affects the trabeculation. So to quantify the contractility, we first use transgenic zebrafish that we collaborate with Dr. Jeffrey Byrne and Caroline Byrne and the MGH. Oh, they actually moved to the Boston Children's Hospital like recently. So they are nicely provide us the transgenic zebrafish line to visualize the cardiomyocyte nuclei. So we use that zebrafish to visualize cardiomyocyte nuclei as a marker, and then we aim to track them and quantify the contractility. However, due to tissue scattering and refractive index mismatching, we have done a little bit more image processing to make each cardiomyocyte distinct. So we first applied the feature enhancement algorithm called the difference of Gaussian, which can be utilized to increase the visibility of edges. So we increase the visibility of edges and other detail present in images. And then we use the watershed, the traditional watershed segmentation technique for segmenting each object by using binary techniques. So compared to the raw image, so if you look at these two images, so this binary image give us the clear cardiomyocytes compared to these raw images. So compared to the raw image, now we can distinguish each individual cardiomyocyte very clearly without merging, without having like this blur boundaries, right? So this video shows the 2 days post-fertilization zebrafish and 4 days post-fertilization zebrafish feeding image. So this is the zebrafish is still under developing stage. So this is ventricle and this is atrium, and this one shows only the ventricular image of the zebrafish with only visualizing cardiomyocyte nuclei. So tracking, so we basically track the 3 cardiomyocytes. So tracking 3 cardiomyocytes as a marker, we quantify the area ratio of the innermost curvature and outermost curvature, and reveals the outermost curvature area have a higher contractility compared to the innermost curvature. And then we see the trabeculae mostly formed initiated from the outermost area and formed the outermost area. But we don't see the trabeculae is formed at early stage, no trabeculae in innermost curvature area. So therefore, the contractility could impact on trabeculation formation. Comparison between control group and Garo morpholino injected group. has a higher, the control group has a higher stretch in both longitudinal, so this stretch level, like lambda 1 is a longitudinal direction, and the lambda 2 is a circumferential direction. So the control group has a higher stretch level in both longitudinal direction and circumferential directions. But a circumferential stretch level is a significantly higher compared to Chiaramontan morpholido injected group. So the circumferential stretch could be a more important factor in trabeculation. And then also we are looking at the TNNT2 morpholino injected zebrafish. So this TNNT, after injecting the TNNT2 morpholino, as I mentioned earlier in the introduction, after TNNT2 gene is knocked out, then there's a lack of contractility. The heart is basically not beating at all. So after we inhibit the cardiac contraction completely, then there's a lack of notch activation with this transgenic zebrafish line. So this green fluorescent shows the notch activation with the TP1 signal. So therefore, the contraction is also necessary to activate notch signaling for cardiac trabeculation. Oh, I'm sorry, my program is crashed. So please. I'm sorry. So my Microsoft has been crashed. So now we want to reveal how biomechanical effect mediate notch signaling for trabeculation. Just for a quick reminder that our hypothesis is combination of endocardial and myocardial notch signaling is required for proper cardiac function with trabeculation. So in this study, we want to confirm whether notch is related in trabeculation or not. So first, we added the DAPT, which is a pharmacological drug that inhibits the gamma secretase, which basically inhibiting the notch signaling. Then trabeculae is not formed. So after we treated zebrafish with DAPT, so there is no trabeculae is initiated, and there is a lack of notch signaling, right? So that means the trabeculae is important. And trabeculae is the signal for trabeculation, and notch signaling is initiated by biomechanical force. So after we lower, so to be more detailed, so after we lower the viscosity and shear stress with the injection of the Chiaramontan morpholino, expression of the notch-related genes are all significantly reduced and did not form a trabeculation. However, after injecting the neuregulin-1 mRNA, we rescued both notch-related genes and cardiac trabeculation. So in here, neuregulin-1 mRNA is from the human cDNA. So do you remember earlier that I mentioned that the large number of cardiac genes are conserved between human and zebrafish? So we take that advantages in this aspect. So we use the human neuregulin-1 cDNA, and then we transfect it and then inject it into the zebrafish to rescue the cardiac trabeculation. After we inhibit the atrial contraction reduce the wall shear stress by using WEA mutant. It's the expression of not related genes are also all downregulated, and we don't see any trabeculation. However, after injection of neuroregulator 1 mRNA, it partially rescues the Notch-related genes, but not everything, but partially. But we see a little bit of trabecular ridges, 1 or 2 ridges were formed. So trabeculae is a partially rescued. And we further inhibit the entire cardiac contraction by injecting the TNNT2 morpholino as mentioned earlier. And so there is no shear stress at all, right? So there's no cardiac contraction, which means there's no blood flow. So there's a zero wall shear stress. And it also induced the Notch-related genes are significantly reduced, and we don't see any trabeculae formed in inner ventricle. And now we are curious what happens if there is no endocardial layer. So we used the zebrafish mutant called CRUSH, which deplete the endocardial layer. But blood flow is still ongoing. Like heart is pumping, so the blood flow is still there, but we only deplete the endocardium. In this case, we also observe there is no trabeculation. And expression of Notch is significantly reduced. It implicates the endocardial layer senses the shear stress in mediating Notch signaling. So endocardial layer is very, very important to sense the shear stress to initiate the trabeculation. So we are still under investigation of cardiac contraction and myocardial Notch signaling. So there is not enough data that I can really show at this time. But so far, from our engineering like technique and tools, and such as 4D light sheet imaging and the biomechanical analysis and deep learning technique, we have demonstrated the hemodynamic shear stress activate endocardial Notch signaling for trabeculation initiation. And then we will later to reveal the combination effect of endocardium and myocardial Notch signaling to design wave-like up and down pattern of trabeculation. So I would like to acknowledge all my lab members and collaborators for this study. And then I really appreciate all the support from the funders. And then thank you. So feel free to ask me any questions, and you have the floor.
Really, really awesome and very interesting work. I like we were talking about before you started presenting, I did a little bit of the embryonic chicken heart formation, and we were looking at the same kind of thing where myocardial contraction is far more important for regulating like Notch signaling and EMT signaling than a lot of people realize. So this is a really fascinating study. Do we have any questions from any of our audience members today? If you're faculty online, feel free to unmute yourself and ask. If you're a student, please pop your question into the chat. That way we just don't overwhelm our speaker.
Yeah, so I'm sorry about that. I didn't finish all the myocardial Notch with contractility. So we haven't concluded that result. So I didn't include that result into this PowerPoint. Will be really interesting. So yeah, if you're interested, just follow my research and I'll keep you guys updates. Yeah. So yeah, feel free to ask me questions. Yes.
Yeah. Hi, Juhyun. Congratulations. That's a wonderful talk. Great technique.
Yeah. Thank you so much.
Yeah. I hope we can collaborate.
Yeah.
One question is that you have a lot of experience in this zebrafish model.
Yes.
So you mentioned that you can obtain the genetic models from your peers, colleagues. So, so I also know that people use this morpholino, siRNA. So can you give us some comments that how you maintain the the genetic lines versus that you can use this morpholino, how efficient you can accomplish that.
Yeah, so recently we are actually receiving a lot of comments and critique because we are using a morpholino. So so morpholino has some kind of secondary effect and it's not specifically targeted. So we are trying to move our research into like using a mutant line, like well-known mutant line, where like we are actually trying to figure out how to do the CRISPR technique to make a mutation, generate a mutant. So that's still, we need to learn about like a CRISPR technique. That is something ongoing project. Yeah, that's actually a good point that I got a lot of comments from recent days from the peers and the colleague that the morpholino or those are not very specific. Yeah, so
you freeze your the embryo, right?
Yeah, so yeah, we are using an embryo. Okay. And then, so I have another project that is the heart regeneration that I'm collaborating with Dr. Hashim Sadiq at UT Southwestern, and then the UC Irvine, Hong Kong, that we are trying to understand the the regeneration mechanism, but it's still very initial stage. Then initial stage, we are trying to figure out how we can utilize with the CRISPR technique. Yeah.
Thank you.
Yeah.
Have any other questions from any of our audience members? Haven't seen any in the chat so far.
Yeah.
How costly to maintain a zebrafish facility?
Oh yeah, the maintaining a zebrafish, I think UAB, they have a nice zebrafish facility.
Yeah,
the maintaining a zebrafish is, I think it's much easier than mice or rabbits, and it has a less regulation. Yeah. So, but still, the zebrafish is still zebrafish. We can do a lot of like nice, cool, like basic science with the zebrafish. But my ultimate goal is we need to find some kind of biological mechanism with the zebrafish and translate into the mammalian animal. So, such as zebrafish, we can just completely remove all the red blood cell in the circulation system. But which we cannot do it with a mammalian animal like mouse. If we remove all the red blood cells, then mouse will die. But for zebrafish, their tissue is very thin in early embryonic stage. So even if we remove all the red blood cell or inhibit the cardiac contraction, so there is no heart is pumping at all, but zebrafish still develop. So it has no problem until like 5 dpf because their tissue is thin enough to get oxygen and nutrition by tissue diffusion. So that is the advantage of using a zebrafish by basic science compared to like mammalian animal. But yeah, definitely we need to move our mechanism finding into the mammalian animal. That's actually my goal. Long-term goal. Yeah.
I was going to ask, I saw you use both genetic and biochemical ways to inhibit the contraction and the shear stress. Have you used anything like tricane, where you basically force more contraction to occur and so you get like over trabeculation?
Yeah, so if I can share so I have some extra slide that I can share. Yeah, wait, I think I probably have the slide. But yeah, so basically we have used, we increased the number of the red blood cell
Okay.
to increase the shear stress.
Okay.
Yeah, but that time, so we basically increased the shear stress by, we increased the shear stress genetically, but interestingly, the trabeculae is still remain same. It doesn't affect the overtrabeculation, hypertrabeculation. So I was thinking about like, I was actually expecting that it could induce the overtrabeculation, but it was not. So I think probably like, you know, so a lot of people are studying atherosclerosis, right? So they said the doctors, you know, the cardiologists, they recommend us to exercise, right? So run and exercise to increase the shear stress, right? So if the increasing shear stress, it affects the human health condition, then it's not good, right? So then the doctor probably not recommend us to do the exercise, right? So I think there is some kind of thresholding point. So there is some kind of thresholding point. If the shear stress is over the thresholding point, that it activate notch to initiate the trabeculation. But if the mechanical force is lower the mechanical force is less than this thresholding point, then it won't activate the notch, and there's no trabeculation. That's my assumption. That's my conclusion. Yeah, I don't know whether it clears your questions.
Yeah, no, that was great. I think that was, yeah, thank you. Let's thank our speaker again. I always say the worst part about virtual seminars is that we can't actually give you applause. The best we can do is the little like applause emojis that are built into Zoom.
Yeah.
But thank you so much. I will see most of you back here next week for our next seminar, but Juhyun, if you wouldn't mind staying on for a few minutes,
sure,
we can kind of take care of a few administrative things.
All right, thank you so much.